to a seat. We're trying to determine if we need any more chairs. I think we might just have a right amount. People are slowly swarming. Don't be shy. Yeah.
Yeah.
I'm making my my aunt sit over my mother's. There's more seats for folks kind of sprinkled about, including in this front row. We've got 2 seats here for anyone who wants to sit down. Well, I think with that, I'll say welcome to everybody. And tonight, who's going to start us off with remarks is Bob Byers, our Executive Vice President of the Fort Worth Botanic Garden.
Welcome, everybody. We're honored to have you join us tonight for this celebration. The culmination of 4 years of really hard work on the Carlquist archives. I want to start off by thanking all of our staff and volunteers who spent many, many hours working on this, and I know that for a fact because they did a lot of the work in the meeting room right across from my office. So I was seeing them in there quite regularly for a long time working on this. We also want to thank the National Science Foundation for their $1 million grant. That support made the project possible. Sherwin Carlquist was a pivotally important 20th century educator and researcher who was pretty influential throughout the world of botanical research. When you see the exhibit, you'll realize pretty quickly just how wide-ranging his interests were, and those interests led to a lot of discoveries in several botanical fields, so he was pretty important. BRIT was really honored in 2018 to receive his archives, and we've also been honored to be able to collaborate with our colleagues from the California Botanic Garden on this project. We've jointly developed an innovative approach to making some critical scientific information in our respective collections available to the public, which I think is critical, and it's really representative and a great example of the direction that we want to take our institution in the future. We want to be building better access and better collections. We want to be collaborating through partnerships to increase the impact of that information. And we want to be providing platforms for researchers, educators, and public entities around the world to learn from one another. And I think this project is going to be an example of a great way to do that. So thank you for being here tonight. Please enjoy the exhibit and have a wonderful evening. Don't forget to go back and help us get rid of all that food in the lobby. And now it's my pleasure to introduce our Vice President of Research and Conservation, Dr. Peter Fritsch. And Peter is going to share more information about Dr. Carlquist's scientific contributions. So, Peter?
Thank you, Bob. Well, as you'll see, the exhibit already does a great job in summarizing Dr. Carlquist's scientific contributions. So here, I want to expand a bit on his ideas around island evolution, while occasionally adding thoughts about what they've meant to me and my own career. So I had the honor and pleasure of getting to know Dr. Carlquist during the 1990s at what was then called Rancho Santa Ana Botanic Garden. You'll see it's now called California Botanic Garden. It's in Southern California. The garden has a joint program in botany with the Claremont Graduate School. one of the 7 Claremont colleges. I'm sure many of you know this. Like all the program's PhD students, I did most of my research at the garden while taking classes at both the garden and the colleges. Dr. Carlquist was a professor at Pomona College and a research botanist at RSA. Sherwin, and all the students, by his insistence, called him by his first name. He wasn't my major professor, but like that of countless students, my scientific trajectory was greatly influenced by Sherwin and his work in plant systematics and evolution. Sherwin was a brilliant scientist whose career encompassed decades of fieldwork, experimentation, and theorizing. He's best known for his work on island biology with a focus on plants, but he considered himself first and foremost a plant anatomist. And most of his publications are on plant anatomy, many of which are co-authored with our former CEO, Dr. Edward Schneider. Even in plant anatomy, which can often be simply descriptive, he nearly always placed his findings in an ecological or evolutionary context. What did the physical structure of plants say about the environments in which they grow, and what are their adaptations? You'll see from the exhibit that Dr. Carlquist focused intently on island evolution. Why islands? Well, islands can be considered a kind of metaphorical living laboratory where evolution proceeds in isolation and many of the complex factors in continental systems are absent or simplified and therefore be more easily controlled for, so to speak. From observations during his many field expeditions, he came to the realization that although Darwin had originated the idea of long-distance dispersal, accounting for distribution of many organisms across the globe, there have been very little in the way of actual observation and testing of this idea up to Sherwin's time. And that's what Sherwin set out to do. Dr. Carlquist's dissertation focused on the Polynesian genus Phycia, in which he surmised that its closest relatives were ultimately to be found on the mainland and members of the genus Bidens, which probably many of you know is the tick seed, the little tick seed. He proposed long-distance dispersal across vast oceanic distances for this by external bird transport, with little prongs on the fruits serving as the means of attachment to the bird feathers. He also invoked Darwin's idea of reduced dispersibility of island organisms for both the loss of prongs of the fruit in some of the members of Bidens and the gain in fruit size in one species of Phycia. In fact, it's probably the largest fruit in the Asteraceae. The Asteraceae, it's a huge family of plants. The idea is that retention of high dispersibility in an island plant would mean high wastage of gametes lost to the surrounding ocean. Okay, so this research led to more synthetic studies on the rarity of long-distance dispersal as the driving force for evolution and isolation, including trends in dispersal mechanisms and the discovery of secondary woodiness. That is, woody stems evolving in place on islands from herbaceous mainland ancestors. Finally, he documented the disharmonic composition of island biotas, the fact that certain taxonomic groups are dramatically over or underrepresented on islands compared to their mainland floras, reflecting this kind of lottery-like nature of islands compared to mainland floras. With this data, he proposed that most long-distance dispersals by birds, either external or internal, with much lower percentage by the physical means of wind or water. And he also theorized that what the evolutionary consequences were to the island organisms with the different modes of dispersal. So Sherwin surmised all of this without any of the modern tools that we have today, including DNA sequence studies. But the studies that have been done more often than not strongly corroborate his ideas. His island research culminated in the publication of 3 seminal books that we have, I think 3 of them we have here. Island Life, Hawaii, A Natural History, and Island Biology, published between 1965 and 1974. I've read them all at least once, and if you consider yourself a naturalist of any sort, I highly recommend reading these, even though they're, 50 years old now, they're still really relevant today. maybe something for the BRIT Reads Club? I don't know.
Yeah.
Not done that yet?
You
Okay.
can nominate him.
Okay. I remember one day at RSA, I was reviewing literature for my dissertation on an intercontinental disjunction, that is, closely related species widely separated by area, when Sherwin saw what I was reading, and he had some rather disparaging remarks about the article. Only later did I realize what the fuss was about. The article is on vicariance biogeography, a very popular topic at the time, and the aim was to strengthen testability in the study of biogeography versus the so-called narrative approach, and claimed that repeated phylogenetic patterns pointed to a common and ancient history of organisms across the globe. Often where continents were still connected prior to their continental drift that we all know of today. It's all well and good, but some of them were invoking ancient continental land bridges across the Pacific to account for the existence of the native Hawaiian flora while strongly rejecting long-distance dispersal. This Sherwin couldn't handle. It was partly fueled by the flora's very different look to mainland relatives, and I can vouch for this, having studied Hawaiian blueberries now for some time. They're different in many respects to the mainland ones, to the point where people have placed them in their own taxonomic group. However, based on data from the molecular revolution starting in the 90s, we can now unequivocally state that the Hawaiian native flora has evolved entirely in place, derived from dispersal across the ocean expanse no more than 5 million years ago, and in geologic time, that's pretty recent. The molecular data also showed that long-distance transoceanic dispersal events worldwide have occurred far more often than the vicariance biogeographers were willing to accept, and have vindicated Sherwin's views. Now, as so often happens, in science, we've reconciled these two rather disparate ideas and actually incorporate both of them into our modern biogeographical models. But swinging back to the blueberries for a bit, our data are very clear that the sister lineage to Hawaiian blueberries is a species narrowly endemic to Japan, of all places, and so it appears that their origin is northeastern Asia, likely arriving to Hawaii from birds, as Sherwin would have predicted. So Sherwin was an excellent undergraduate student mentor and engaging lecturer, and although I was never able to take a biology class from him, I did take a botanical German class from him, interestingly enough. He was, I think, fluent in 5 languages. I even kept my textbook right here. And at the time, knew it was $39.70. Now it's $150. I looked it up today. Crazy. Yeah. So now I can say words like das Pflanzenreich and Pflanzenfamilien meet das Vertrauen. Maybe I can't say it that well. For the class, I was tasked with translating the Pflanzenfamilien treatment for the family Hydneraceae, a bizarre group of holoparasitic plants whose closest relatives are now considered to be in the family that includes wild ginger, and that's what this is right here. We've got a volume, of course, in our library. I think Dr. Carlquist's main legacy derives from his ability to bring into focus the great potential in studying island organisms for a more accurate understanding of evolution, and doing it in a way that was both compelling and easy to understand. His work also has had key implications for conservation, because so many of island species are now rare, threatened, or even, of course, extinct. It might surprise you to know that many of the island plant groups that he studied haven't been looked at again with molecular data since, so there's a lot of work still to be done. This was certainly the case for the Hawaiian blueberries. So in 2018, I steered a graduate student toward this group. So with Sherwin's fundamentals as our guide, we've been corroborating the hypotheses that he set forward and making new discoveries. I'm confident that Sherwin would be very pleased with our progress. As for Sherwin himself, he was somewhat shy, according to colleagues, sometimes difficult, but in my experience, a very kind man, always willing to share his time and his knowledge. He had probably the most comprehensive personal collection of 20th century classical music probably in the world. I'm not exaggerating. He had thousands. He was an excellent chef. Oddly, I remember that the first time I ate capers was at Sherwin's place. I didn't know what a caper was. I grew up in Iowa, we don't have vegetables there. He was an by rights, he should have been elected to the National Academy of Sciences. Back then, at least, it was kind of club, and you had to be nominated by other members. It's still a little bit that way, too. But I was thrilled when in 1996, the year I began my first job at the California Academy of Sciences, I was able to host Sherwin during his visit to receive its prestigious Fellows Medal for scientific achievement. So I hope this has given you some idea of the impact of Dr. Carlquist's creative yet analytical mind, something to note as you take the exhibit into account. With that, I'll turn it over to Ann Nino, our BRIT librarian, who will tell you more.
Good evening, everyone. My name is Anna Nino, and I'm the librarian here at the Botanical Research Institute of Texas. Thank you for joining us this evening at the opening reception of Expeditions Through the Lens, Botanical Photographs of Sherwin Carlquist. This exhibit offers a view into this renowned American botanist's career, the extensive photographic record he produced during his field expeditions, and the impression he left on the fields of wood anatomy, systematics, and island plant diversity. In talking about our man of the hour, I'll teeter between calling him Sherwin and Carlquist. Sherwin was born in Los Angeles, California on July 7, 1930. Growing up in the suburbs of LA, he visited the Huntington Garden on weekends, which is where his enchantment with the forms of plants began. Awarded a science scholarship his senior year, off Sherwin went to college, earning his undergraduate, then his PhD in botany at UC Berkeley in 1952 and 1956. Carlquist's postdoctoral studies led him to Harvard, where in a letter to Peter Grady Webster, he called his 2 years there rather dull, recounting a flood that racked his basement office, and he bemoaned his lopsided office chair. Sherwin didn't stay in his rickety Cambridge chair for long before hopping back to California, where he accepted a teaching position at Claremont Graduate University and Pomona College. Plus, the position came with a spiffy lights microscope and attractive office furniture. Eventually, he became the plant anatomist at California Botanic Garden, which is now called, or was once called Rancho Santa Ana. During his tenure at California Botanic Garden, Carlquist traveled extensively, his research propelling him to over 26 countries, mostly Pacific Islands. With travels fueled by grant funds and his insatiable force of curiosity, Carlquist was asking questions about how plants adapted to their environments, how they distributed to these remote locations, and how certain species then evolved depending on various factors. Through the world map hanging next door, you'll appreciate the extent of his travels and how many specimens he collected at each vicinity. To get to these corners of the world, the aviation capabilities to reach these remote isles had to exist. He flew by plane and jet, drove rental cars to collecting spots, embarked ferries, and carried himself by his own 2 feet, except for the occasional trips and falls. During one of his falls off a Tahitian trail, actually, he landed next to a new species of scaevola. So he fell for scaevola. He often embarked on expeditions to these unknown terrains with other botanists and locals. And in one of the field expeditions photographs, you're going to see local guides from Papua New Guinea sitting, looking out over the Sarawak Range in Papua New Guinea, and they will be holding a basculum, which is a metal case for storing and transporting specimens, as well as a canvas bag. Carlquist studied plants from every angle and at every scale, snapping aerial photos from 10,000 feet in the air, then on the ground of a plant in its entirety, as well as close-ups to feature specific anatomical parts. He would record information about the plants he collected in his field notes, and we've included some select field notes in the display cases. They're a treat to look at in all their forms, including the Darling Red Pocketbook Journal to a scrap piece of paper. Once collected, he would preserve specimens in multiple ways. Leaves, fruits, seeds, flowers, and pollen were pressed on herbarium sheets or contained as fluid preserved samples. Branches would be cut into wood samples. Specimens were then shipped by freight back to his lab in California. For pickled specimens, Carlquist would often just use whatever jars were on hand. It seems obvious to me that another character figured heavily in Carlquist's life and in this exhibit, and that's his photography itself and the equipment he used to produce such lucid imagery. After his professor, Marion Cave, trained him on how to develop film, it was a love affair with photography from then on. Sherwin started with a 35mm camera, but made the switch to Hasselblad in the 60s, owing to the Hasselblad's high image quality and its modular capabilities. He customized his camera even further with Zeiss and Leitz lenses for his high magnification photos, such as the vibrant ultra close-ups that you'll get to enjoy in the UCU section. The combination of the camera and lenses allowed him to get up close and personal with the interior structures of plants, which he would reveal by slicing into cross-sections. Beyond observing the plant world, Sherwin took advantage of his travels for excursions to temples, theater performances, and festivals, demonstrating his interests in art, architecture, and culture, snapping photos of these outings. His documentation of slices of the world over 60 years poignantly reveal the world's transformation on biological, ecological, and cultural fronts. Sherwin's approach to understanding the natural world spilled across many botanical subfields, oft explored separately from one another. He integrated the perspectives and practices of taxonomy, ecology, field botany, and plant anatomy, layering them into an expanded outlook. He gleaned valuable insights from physically witnessing plants in their habitat standing right next to them, but he also highly valued the over 11,000 physical plant specimens he collected, which served as references for new species determinations and formed the basis for the ideas he explored in his publications. With his exceptional memory and ability to pick up on patterns within and across plant families, Sherwin tackled plant discovery across taxonomic lines, collecting species that until then had been poorly studied and describing 63 new plant species during his career. As Peter mentioned, Sherwin worked and published extensively with Dr. Edward Schneider, previous CEO of BRIT and a mentee of Sherwin's, which led to Sherwin donating his field photographs, field notes, and index envelopes to BRIT Library in 2018. Since his biological and archival collections were now divided, California Botanic Garden and BRIT now teamed up with the proposal to digitize his collection of nearly 200,000 items, connecting them across their distinct collections platforms through bidirectional hyperlinks. The shared hyperlinks, or what the Carlquist team calls linkages, connect the original plant specimen that he collected with its corresponding field photograph, field note entry, and or index envelope. And if you don't know what an index envelope is, check out the display cases next door. This linking approach stems from a conceptual model called the Extended Specimen Network, or ESN, coined in 2017 by ornithologist Michael Webster. The ESN model comprises 3 concentric circles, or extensions, with a specimen situated at the core as the irreplaceable primary source from which other versions of the specimen or auxiliary data connected to the specimen and collecting event are derived. By linking these otherwise siloed materials, BRIT and CalBG have laid a red carpet for researchers to better understand the rules that govern how organisms grow, diversify, and interact with one another, and how environmental change and human activities may affect those rules. This project cements how these collections, both biological and archival, are the raw materials that form the foundation of new knowledge. New knowledge is created by building on existing knowledge, and existing knowledge lives in these objects that libraries, herbaria, and natural history collections curate, maintain, preserve, and make discoverable. Carlquist's collection materials and ancillary data, especially when understood in their interconnected context, allow us to ask more interesting research questions. This project has also revealed to the Carlquist team at both institutions the community infrastructure necessary to link objects successfully across distinct platforms. Linking objects takes qualified individuals who can critically assess whether there are connections between a plant and a photograph of the plant. It takes willingness on the part of collections managers across domains to collaborate and attempt to understand each other's terminologies and best practices. It takes a technical capacity within distinct databases to connect to outside digital assets. It takes new vocabularies and relational terms that can coexist between platforms and across metadata schema. The ESN offers research so much, and its complexity necessitates sustained attention, much like the quality that Carlquist exhibited throughout his life. Now, time for acknowledgements. Thank you to the U.S. National Science Foundation for making this project possible. I also want to introduce our BRIT Carlquist team members who have spent hours upon hours with this collection, digitizing it, understanding it, and training others on how to do the very same. If y'all could please stand up when I say your names. Krishna Shenoy, digital archivist. Sam Epberg, digitization technician. And Jason West, director of biodiversity informatics. To these three individuals in particular, I have cherished the opportunity to work with you in developing this exhibit, drawing on your unique skills and talents and your dedication to honoring Sherwin's legacy. A shout out also to our teammates in Claremont in California Botanic Garden, and specifically the lead PI on this grant, Maren Nazaire. Without her and my predecessor, Brandy Watts' coming up with this ambitious notion to pull something off like this, we wouldn't have this project idea. And thanks are also in order for University of North Texas Libraries and Symbiota Support Hub. Oh, where am I? The two repositories hosting our digitized images. From our education department to the herbarium team, from marketing to facilities, thank you for your support in bringing this exhibit to life. And I think most importantly, I'd like for anyone who has volunteered on this project to please stand right now. Huge sum of gratitude goes out to you, our practicum students, interns, and community volunteers. From 2022 until now, this work would be nowhere without your commitment. In conclusion, I want to thank you all for being here and supporting the work we do to preserve and celebrate botanists, horticulturists, and other plant-loving people's legacies. May Carlquist's ravenous appetite for learning, questioning, and documenting inspire your own hungry pursuits. Thanks. And please enjoy the exhibit. It'll be here until September, so invite other people to come out. We'll be here until 8 p.m., maybe a little later if that just happens. But please also take food, talk to each other, and enjoy. Thank you.